to our webinars. Welcome to our webinars on food and microbiology. A special thanks to our international guests from the USA, from Canada, and from Australia, Professor Katouli, and also all other speakers. Dr. Rahimi, can you hear me now?
Yes.
So you can continue and handle because you were gone for a few minutes. So I started. Then you can invite Professor Katouli, our guest professors in our previous Congress, as the first speaker.
Yes, now I want to say hello to everybody and welcome to our panel in the 22nd International Virtual Microbiology Congress. It's my honor to invite Dr. Katouli for 15 minutes and he can present their presentation. Dr. Katouli, are you hear me? Dr. Katouli?
Unmute now. Yes, I hear you.
Yeah.
And
Thank you very much.
Yeah, because you are in control and I couldn't sort of let me share my screen.
Please introduce yourself, Doctor.
We'll do that in a minute. Do you see the full screen?
Yeah.
You see my full screen?
Yes,
No.
we can see your full screen mode, but can you make it duplicate? I mean, in the display setting, please make it
I'll do
duplicate.
that.
Yeah, thank you.
Okay, thank you very much, Dr. Rahimi Fard and Dr. Faizabadi for organizing this beautiful and most productive, I'm sure that it is, seminar that is being done from all over the world. This is the technology, and thanks to sort of technology made everything available. My name is Mohammad Katouli. Excuse me. I've been working all day, so my throat is really, I've been teaching actually, is dry, so I need a little bit water. I am working at the University of the Sunshine Coast as a sort of a teacher as well as researcher. My group has been working on interaction of microorganisms with gut microbiota. And one of the important aspects of these gut microbiota are probiotics. And I'm going to sort of present to you small amount of the sort of the big project that we have been doing on probiotics and what these probiotics that nowadays we call them live biotherapeutic products do to human gut. The whole history of probiotics came from Eli Meshnikov early in 20th century. And this particular Russian scientist, who was actually what I would call him the father of immunology, he noticed that peasants and farmers in Bulgaria, they have a sort of this longevity of life. And he assumed that this is because of the immunological responses of the body. But then when he went and lived with these people, he noticed that the main food of these people are fermented milk in the form of yogurt. And he started looking at the yogurt. And of course, nowadays, we know that yogurt has a number of bioavailability, far better than milk. But the main ingredient of the yogurt was bacteria that he noticed. And he brought it to the attention of the scientists in the world that these bacteria most probably have something to the health of the human being. Of course, he was given a Nobel Prize in 1908. But later on, it was in 1965 that two scientists, Liley and Still, they sort of coined the name probiotics as opposed to antibiotics to show that these are live microorganisms with beneficial effect on the host. Now, the full definition of the sort of probiotics came later on, that nowadays it's been accepted by many scientists around the world that probiotics are referred to live microorganisms that when they are consumed in adequate number, and that's very important, confer a health effect on the host. Since the coining of the probiotics as groups of bacteria that they have a beneficial effect on the host, a lot of research was done to see what is the interaction, what is the consequence of interaction of these probiotics, mainly in the gut. Later on, a little bit work was done on the vaginal microbiota and the type of lactobacilli that they are present there. But the main work was done on gut and the interaction of these bacteria alone or in combination with the sort of the normal microbiota. Now, one of the things that is important here is that gut microbiota is made of thousands of species, at least more than 1,000 species are there, and the interaction of these microorganisms establishes a very stable ecosystem that cannot be easily changed unless there's some probiotic or something which is very devastating to the life of the other microorganisms. But majority of them are in a very small number. So the life of the probiotics and the colonization of the gut, their competitive exclusion of the pathogen, as well as immunological response of these microorganisms on gut epithelium has come to the attention of many scientists. For instance, in my group, we are working on the regulation of the sort of certain genes that they are involved in production of the sort of or overproduction of the sort of gut epithelium at the crypt level of the sort of gut. We are trying to use probiotics to control the overexpression of these genes. But what is important here is that a number of companies, they have taken advantage of these good bacteria and they have formed either these bacteria as sort of alone, such as Lactobacillus GG, initially developed by Golden and Goldbach. And then number of companies, they started using a combination of different lactobacilli with the hope that they have a far better effect than single lactobacilli. For instance, one of the products called Probiotic 14 is made of 14 lactic acid bacteria, including lactobacilli, including Streptococcus, Bifidobacterium, and so on. Later on, another group or another company, they started sort of making a raw probiotics. And these raw probiotics was made of 34 different lactic acid bacteria. All are designed to give a sort of a good stability and of course, better health to the sort of the human being. Now, one of the things which recently over the past 6 years has come to the focus is that there are certain groups of lactobacilli or probiotics, if you like, that they are alone or in combination can be used to alleviate or treat the sort of certain diseases of the GI tract, mainly inflammatory bowel disease, including Crohn's disease, ulcerative colitis, and colorectal cancer. Now, these probiotics, they have been defined under certain names to distinguish them from those probiotics that they are associated with food. And nowadays, the term live biotherapeutic products or LBP used by American, beg your pardon? Yeah. And in Europe, they are also using next generation of probiotics. So, despite the fact that these live biotherapeutic products have been introduced, not very much work has been done on the interaction of these combination of the cocktail of these probiotics on human gut epithelium, especially those in terms of alleviation of the disease, such as inflammatory bowel disease. And of course, if you want to work with IBD, you have to either use probiotics in clinical trial, which is very difficult, and there are a lot of ethics are involved, or you can use the test in laboratory if you develop a good model. So in many studies have shown that one of the microbiological or etiological of the sort of inflammatory bowel disease could be E. coli, and certain E. coli species have been commonly found to be associated with the mucosal membrane of patients that they have IBD and colorectal cancer. These E. coli strains have been characterized, and they have been shown to adhere and invade the sort of the gut epithelium, and they survive and replicate within the macrophages. Besides, they have a certain toxin that we call them genotoxin. These are characteristics that you cannot see them in normal or other pathogenic E. coli. And for that reason, they have been termed as adherent invasive E. coli, or AIEC for short. So we were very much interested to test few of those probiotic strains that they have been claimed and been shown that they met all the sort of characteristics of probiotics, and we wanted to see whether they work alone as a sort of product that can alleviate the or competitively exclude AIEC in a model of the gut, and also look at their combination as it's been suggested by many pharmaceutical companies. So at the absence of a sort of a clinical trial, we decided to use gut epithelial cells. And gut epithelial cells that we normally use are CACO2 cells or HD29 cells. Now, these cells, they have certain characteristics, and certain sort of characteristics are actually missing from these cell lines. For instance, the CACO2 cell, which has a sort of tight junction, as you can see them here, they have a sort of producing macrovilli, and they have certain enzymatic activity, but they do not produce mucin the way that they do in the gut. On the other hand, HT29 cells, before being treated with methotrexate, they showed that they do not have a very strong tight junction, and they do not have macrovilli. So a mutation of these cells with methotrexate produce a type of cell line that we call them HT29 MTX. And here you can see that this particular cell line, they act like a goblet cells in the human gut epithelium. Here is a CACO2 cells, as you can see them, when we stain them with Alcian blue, which is specifically colorized. The mucin doesn't show any mucin on top of that. HT29 MTX alone, you can see that it produces a lot of mucin, as it's been shown in this color. And then when you join these two as a co-culture at a ratio of 9 CACO2 cells and 1 sort of HT29, it also produces mucin. So in fact, here we have developed a sort of a kind of model for the gut that is not only have all the characteristics of the gut epithelium, but also produces mucin, which is very important in interaction of the bacteria with the gut. Now, to test these probiotics, we also looked at another factor, and that is looking at whether these probiotics can be competitively excluded by these probiotics, or the probiotics can sort of prophylactically sort of inhibit interaction of these bacterium, which is AIEC, E. coli, on the gut epithelium. So we use two sets of cells that we call them co-inoculation to represent competitive exclusion, and prophylactic, which is a pre-inoculation. And here is that to show that AIEC and lactic acid bacteria, they adhere to these cell model. We use them, and we use the scanning electron microscope and normal sort of to show that they adhere. And of course, here we show them that these bacteria, these put on this culture, they invade that. So this is the result that we obtained. You can see that these E. coli AIEC can colonize almost 60% of the co-culture. And of course, lactic acid bacteria alone, right, they also do the same things. So they can colonize the entire surface, and we show them with electron microscope that they colonize them. But when we started putting this thing together, which as a competitively to see whether they competitively exclude AIC, we saw that each individual probiotics, they can do that, except this one, rotaride, that does not reduce the sort of the colonization of the sort of AIC in these cell lines. And of course, we didn't see very much effect with a combination of for lactic acid bacteria. When we looked at the adhesion of these bacteria alone, or at the presence of lactic acid bacteria alone or in combination, we saw that there is a sort of, when we use them as a co-inoculation, there is some reduction. And of course, when we do this thing at the pre-inoculation, sort of these green ones, this gray one, you see them, the pre-inoculation reduces dramatically, far better than co-inoculation. We were a little bit worried about that, why this is the case. And when we did the invasion assay to see that these bacteria invade the sort of epithelium, as it's been shown in electron microscopy, then we saw that yes, AIEC alone can invade the cells. And of course, when they are combined or pre-inoculated with probiotics, there's a tremendous reduction in all of them. The most effect was when prebiotics alone or in combination were used as a sort of a pre-inoculation, which suggests that if you are going to use these probiotics either alone or in combination, you have to present them as a sort of a prophylactic measure rather than competitively excluding pathogen or get rid of the problem associated with this particular E. coli. And here you can see that in pre-inoculation, we have a sort of far better reduction in the sort of immediate adhesion or invasion of these bacteria as opposed to co-inoculation. To make things short here, this is a sort of our conclusion that all 4 strains of probiotic that we used alone, they had a sort of good effect when they are sort of used as a both co-inoculation and pre-inoculation. However, when we use the sort of combination of these 4, we didn't see very much sort of effect to improve the already reduction that we see with individual probiotics. But what we could see that, and it was very clear in all experiments that we did, was that pre-inoculation always far better in controlling the adhesion and invasion of these bacterium, which is normally associated with a sort of inflammatory bowel disease. And this suggests that if you're going to use probiotics or what they call them live biotherapeutic products, the best model that we can use is that to give them as a prophylactic measure rather than as a treatment measure. Although this is not a very complete sort of experiment, we are doing some more experiments to improve the method, including addition of the secretory IgA, which is commonly found in the gut, and also at the presence of the normal microbiota. But at the end of the day, there are 2 things missing here. And before we get those things done, we cannot make a full judgment on that. And one of them is that to create a more powerful sort of combination of a strain individually and in combination, and also test them in clinical trial. Under that condition, then we can see that live biotherapeutic product can work to alleviate or treat sort of diseases of the gastrointestinal tract. And at the end, I would like to thank my colleagues in Iran, Dr. Pushafi, Dr. Talebi in Pasteur Institute, and Iran University of Medical Science, respectively, and my colleagues here at USC, Dr. Kubala, Dr. Haji, and my students, George, Albert, Christian, and Chaimika, that they contributed to this work, which part of that has been presented in this seminar. Thank you very much indeed.
Thank you, Dr. Katouli. And now, Dr. Abdullahi, can we invite Dr. Ruth? Thank you, Doctor. Thank you. If any question everybody have, because the time in the local time of Dr. Katouli is very late in the midnight, so you can ask him, and after that we can say goodbye to Dr. Katouli. Now we have 5 minutes for any question from Dr. Katouli. From one of excuse me, I can't see.
Chats to see.
We have some question. In the text, we have some question. Excuse me, Dr. Abdullahi, could you please read the question? I'm with mobile.
Yes.
Please.
Is it necessary to stop using probiotics after using them after a while?
Well, actually, if it's going to be used for treatment, then of course you have to continue all the way. But normally probiotics as such, they are supposed to be there because there's not very much work to show that after a while, these probiotics, they can really colonize the gut and reside in the sort of the gut, because gut is a very sort of a milieu full of changes every day that we eat different types of food, different material getting there, and these bacteria are definitely affected, especially they are outnumbered by groups of bacteria such as Bacteroides, Bifidobacteria, and so on. But it looks as if that if you are using probiotics for treatment, then you have to continue even while after the sort of the alleviation of the disease occur to make sure that there is no recurrence. In terms of using them for health effect, my guess is that you have to use them constantly, and there are capsules that you can take them one a day or one every second day. Any other question?
Yes, Dr. Abdullahi, we have another question. Please read.
No.
Mr. Moradi, I think.
Sorry, we have another question from Dr. Warriner. Dr. Warriner, you can unmute yourself and ask your question.
That's good. Nice presentation. The question I was going to ask is that obviously lactic acid bacteria produce bacteriocins, which could affect other lactic acid bacteria. So, especially Rootii, that's obviously a very potent bacteriocin. So, do you think these combinations of the lactic acid bacteria could actually counter each other and therefore produce a more negative effect than a synergistic effect?
I totally agree with you. And one of the studies that was done many years ago, it was a sort of combination of 6 lactic acid bacteria. One of them was actually Rootii, and it looks as if that the production of bacteriocin affects other groups of bacteria. In that study, actually, it had a negative impact on the sort of the health of the sort of people that they took this combination of a strain. Since then, there's been a lot of speculation as to what combination of the lactic acid bacteria we should use to boost the sort of the effect rather than to inhibit their effect. You're right. I mean, they produce bacteriocin and some other sort of products like that they can kill on or inhibit the growth of other lactic acid bacteria or have a sort of negative impact. I totally agree with you. Yes, we have to find the right groups of lactic acid bacteria. They synergistically boost the effect rather than, yeah.
Dr. Moradi, you have microphone. Please ask your question.
Yes, thank you. Thank you, Professor, for your great presentation. I have a question. You said that we can use the cell line model to stimulate the gut environment, since there are a lot of factors, I mean, host factors that can affect everything. I mean, biochemical, that the produced by the microorganism in the gut. So how much the results of this cell line model can show the real effect? I mean,
Well,
instead
yes,
of using the gut environment.
Yeah, I agree with you that the gut sort of a milieu is a sort of a very complex milieu, right? And there are thousands of species of bacteria, and they interact with each other, and they interact with the gut or the host. The thing is that when you are doing experiment like that, you have two choices. Either use a sort of human being as a model that is practically, ethically very difficult to do that. And of course, many studies using probiotics, they have shown contradictory results because the nature of the human being that they have tested or the genetics or the race have been sort of varying very much. On the other hand, the only choice that is left with us is that to try to establish a gut model that is closer to what we normally do. If you're going to use one cell line, like as Kakutu cells, you are dealing with few characteristics of the gut, but not the entire gut. A number of studies nowadays, they try to use the whole microbiome of the gut and put them on the sort of C cell line. And one of my PhD students right now is doing this thing, which is very difficult because the results are not reproducible as we thought it would be, because every time that you collect the gut microbiota and add it to your cell culture model, you get a sort of a different result. So the only way that we can look at a sort of more reproducible result is that try to use a cell culture, but we have to improve. So it's improvement. Now we are using a lot secretory IgA, and we are now adding a number of enzymes. We are trying to extract the sort of we get a gut microbiota, we wash them, and we use the extract and use them together with the medium to see what would be the effect of these material produced by the sort of other groups of bacteria on our cell model at the presence of probiotics. So it's a very difficult question to answer, right? Because the gut microbiome is a very complex milieu. And at the absence of the human sort of volunteer, which again, it has a certain problem itself, we cannot use them, and we have to rely on cell line. But we have to improve our cell culture model to get it.
So you're saying that we can have some strategy to improvement our experiment by adding some factors, host factors. But is there any strategy to provide bacterial interaction? For example, adding some bacterial to provide the gut environment?
Yes, we can do that. We can use individual bacteria, particularly those that are high in number, such as Bacteroides, right? And of course, the condition of the gut is that it's a sort of a multicellular. We have to not only enterocyte, we have a sort of a goblet cell, we have an end cells, right? So these are the things that we cannot really provide them as a whole in a laboratory, but we are trying to use them as close as possible. Adding few bacteria may or may not really give us a sort of better result that we think it would. Otherwise, the best thing, of course, is that to use the whole microbiome and add them to the cell culture, which is a very difficult thing to do. And believe me or not, we've been doing this thing for more than a year, and we don't get a very reproducible result.
Okay. So, but it's possible. So you have done it yet.
It is possible. It's a matter of development, and it takes some time. And our knowledge is sort of getting better and better about the sort of the whole ecology of the gut microbiome, as well as the sort of the way that these cell lines can be improved to mimic what is in the gut. It's a matter of.
Thank you, Dr. Katouli. We have another question in the chat box.
You're welcome. And yeah, good luck.
How did you measure the number of bacterial probiotics, CFU, on ml that can be applied in cell culture?
Well, in adhesion assay is very easy. Then you can sort of inoculate them, spike the sort of the culture with bacteria, and then you wait for 1 hour or 90 minutes, and then you get rid of non-attached bacteria. You fix the cells and assay them, and you can count them on a cell. And we normally count 100 cells randomly, and we look at the bacteria adhering per cell, and then we make a mean value plus the standard deviation. When it comes to invasion, we look at the sort of the bacteria entering the cells, and then we kill all the bacteria which are outside the cell with gentamicin, and then we take the cells, we sort of lyse them, and then once we lyse the cell, then we release the bacteria and we count the number of bacteria on normal agar plates.
Okay, another question is, what initial OD measurements would you prefer for the cell suspension if you want to perform co-aggregation for the bacteria and yeast combo? Do you believe it needs to be the same initial amount of bacteria and yeast?
We normally use a multiplication of infection of 100, which means that there are 100 bacteria per cells in the sort of the cell culture model that we have. And we count the number of cells initially by taking few sort of wells, drip size them, we count the number of cells, and we know that how many cells on average we have per well, and then we adjust our bacterial suspension to give a multiplicity of infection of 100, which is 100 bacteria per cell. And this is a sort of something which has been used in many studies internationally.
Okay, thank you. It shows that our question is finished. Dr. Rahimi Fard, could you continue? Dr. Rahimi Fard?
If it's okay, I'll leave the group because it's too late now. And
oh, thank you. It is appreciation. We are
Thank
appreciating.
you. Thank you, Dr. Katouli. Thank you very much for your valuable presentation. I hope you safe and healthy and happy for every day. Thank you very much.
Bye.
Bye. Bye. Thank you. So now I would like to invite Dr. Ruth. Are you ready for your presentation?
Yeah.
Please share the screen, please. Thank you. And I would like you introduce yourself to everybody. Thank you very much.
Hello, everybody. I'm Dr. Ruth Eden from BioExpert, and I want to talk to you a little bit about evolution of food pathogen detection. Somehow my thing is not working. Okay. So for the last 5 decades, from the establishment of the traditional culture media, most laboratories are still using a standard methodology because it is sensitive, inexpensive, and does not require capital expenditure. However, it is laborious, time-consuming, and most importantly, it's very slow to give results. As we can see here from example of Salmonella, if we take a sample, we do a 1 to 10 dilution in enrichment broth. After 24 hours, we transfer it to another selective broth, then to several plates. And if there are no calamities on the plates, we decide that there is no Salmonella, but in most cases, there are some Salmonella or some other organisms on the plates, and it takes 5 days. And if you have an outbreak, a food outbreak, 5 days is way too long to know the answer. If you have product on the line, it's also way too long for it to take so much time to get to detection. So as a result, many people in many countries have worked on a development of faster methods for detection of pathogen. And I had a great vantage point to look at those development and participate in some of them. And the timeline that you see here shows the time when the method came to market, not the time when people started working on them. I remember still in 1990 when Ellen Rombach came to DIFCO and showed us the first chromogenic agar. We ourselves worked on immunoassay and others work on molecular and so forth. And I want to go through the process of how those methods were developed and what advantages do they bring. So we'll start with chromogenic media, which is a selective differential agar that uses chromogenic substances to color the colonies in different colors so we can easily distinguish between the target and non-target. In this particular example, the interfering organism can be in blue, whereas Salmonella have a purple pinkish type of color. Some of the dyes that are used for Salmonella detection are X-gal, which is 5-bromo-4-chloro-3-indolyl beta-galactopyrimidase. And it is not used by Salmonella, but rather by other organisms to create blue colonies. And in order to get a color for Salmonella, beta-glucoside is used. And so we have Enterobacteriaceae in blue color and Salmonella in a red purplish color. There have been many chromogenic medium developed over the years. Even the Salmonella methodology have been improved over the years by using, for example, magenta cap instead of X-gal in order to get a crispier color for Salmonella, and most importantly, in order to detect Salmonella that have atypical biochemicals such as typhi, paratypho, cholera sue, and Gallinarium. So moving along to immunological method, all immunological methods still require the pre-enrichment step. So we don't save a lot there, but the assays after is quite faster. There are 3 big categories of immunoassay. One is a microtiter plate, 96 or 92 wells for doing ELISA. We have lateral flow methodology, and we have immunomagnetic separation, which is really not a detection method, but rather a concentration method to allow other methods to be done faster. And all immunoassays are based upon antibody-antigen type of reaction, and well-selected monoclonal antibodies are the key to the methodology. And just kind of a reminder, this is a lateral flow where you deposit the sample on one end and it moves to the other end. If the target organism is present on the conjugation pad, it gets conjugated, and we have a test line and a control line. Here we see only the control line, and here we see the control line and the test line. The immunomagnetic separation is based upon using magnetic bead coated with monoclonal antibodies, and if we add them to a tube that contains the bacteria, other bacteria, and debris, the beads connect with the bacteria, and with a magnet we can pull them to the wall of the tube, pour the liquid out, and have concentrated bacteria and beads at the bottom of the tube. And those beads can be used as a first step in other assay, let's say molecular type of assays. The molecular assays were developed over many years. It took 15 years from the time people started working on it until the methods came to fruition as a method in food. And it started with simple PCR, multiplex PCR, qPCR, and a variety of other methods. Basically, this type of method takes the sample, pre-incubate the sample overnight in a broth, and then the sample is centrifuged, cell are lysed, incubated with probes, and entered into the PCR type of machine. So we can see the method, this method brings us to an overnight 24-hour type of time frame, and it is also more specific and more reproducible than the standard type of methodology. And until now, PCR is the most common method used, but there were many advantages done over the years in order to bring us to where we are today. For example, using messenger RNA instead of DNA was used in order to avoid fragments of dead cells from detecting in the system, and also use. Okay, let's move along. Okay, and the next method I want to discuss is phage-based type of methodology. As you can see here, let's say it's a cell of Salmonella, and we infect it with appropriate bacteriophages, and as a result of it, the phage injects their DNA into the cell, the DNA multiplies in the cell, and eventually the cell lyses and bursts, and the DNA particles from it get into the solution. And there were phage work done for detection of a variety of Salmonella, Listeria, E. coli, and many other organisms. Most phage methodologies are not used by themselves. They really are used to gain specificity and to fasten the results. If our goal is to get the detection time in 8 hours or within one shift, as opposed to overnight, the only way is to accelerate the time it takes us to get to the numbers required for the, let's say, PCR type of assay. So if we use a phage, the multiplication of the phage DNA inside the cell makes it much faster, and we can get results within 8 hours. There are literally hundreds of articles in the literature describing phage methodology, but I found that the reagent stability is difficult to commercialize, and maybe that's the reason that we don't see any significant mass production of phages for use in detection of pathogens. Another method that got lots of research is biosensor devices, and they consist of two elements: a bioreceptor, which can be a ligand, enzyme, antibody, peptide, or whole cell. And if we take a food product that contains pathogen and interact with one of those bioreceptors, a signal is being created that can be amplified for detection by a transducer. So the biosensors are really capable of converting a biological signal into an electrical type of signal. And the bioreceptor is responsible for recognizing the target pathogens, whereas the transducer can convert the biological signal into a measurable electrical signal. The advantage of biosensors is that they are rapid, sensitive, low cost, and most of them are small. You can handheld them or attach them to a smartphone, and they can be used directly in the field, so they don't need a laboratory. And different transducer configuration have been reported for years in the literature. And but on the other hand, it's difficult to ensure the reliability and stability of the biomaterial that come with the biosensors. So although, again, lots and lots of research have gone, and it is a fascinating area from the point of view of what supposedly it can do, it have not come to fruition as far as I know. Now, one thing that everybody wants, and I mentioned it, is to get the detection time within an 8-hour time frame, meaning that you can do it within one shift. And in order to do that, one other thing that people tried is to find a way to concentrate the microorganism and separate them or purify them in order to get the numbers high enough so you can put them into a PCR type of assay and get the result immediately without the need of a broth multiplication. And for example, here you take a food, mix it with kind of a broth, and then you can centrifuge it, filter it, or use immunomagnetic separation. And after that, you can use nucleic acid, immunological method, or biosensor. And this is a good review paper that describe all those various options that people tried in the literature. Now, the latest trend is to detect multiple pathogens in a single assay. Won't it be fantastic if we don't need to know in advance if we have a Listeria, a Salmonella, an E. coli, or if we have a bacteria, a virus, or a parasite? Everything that shouldn't be there, any pathogen that shouldn't be there, should detect in a single assay. And many people view that as a current goal, and there are in the literature a lot of studies, or at least several studies that describe such methodology. I'll describe only one because we don't have a lot of time, but Dr. Del Agunremi from the Canadian Food Inspection applied genomic in order to detect multiple pathogens. He basically amplified the DNA in the sample and then sequenced all the DNA. And the key to the method is, the key to the method is the Cosmo ID proprietary technology that take all the information. It's really a software packet that take all the information and put it together as a segment of DNA in order to decide which organisms are present in the sample. The work they've done is really preliminary, but they use spiked food samples that were inoculated with viruses and bacteria and other pathogens. And he also took Salmonella samples that came from recalled samples from the Canadian government. And basically the very preliminary data that they showed is that if he had Salmonella and Cryptosporidium, he had both of them, but there was no virus. If all of them were present, he detected all of them, and he did different ratios of the pathogens in order to see that he could detect all of them. Also, he used all the samples that came from Salmonella infected recall samples, and in all cases he could detect the Salmonella, and in some cases with more specificity than the original study had given to the samples. So after kind of this very quick review of the method that have been developed, the question is, where are we today? Basically, after 3 decades of intensive work on the detection of pathogen, the evolution of many modern methods that are fascinating, the question is, who is using this method and how much use is being done of those methods in the field? And obviously the proof of the pudding is in eating it. So Bob Ferguson from Strategic Consultant have done a large survey of food companies in the US to see which methods are using in microbiology. And at least to my surprise, he found that 53% of the companies were using PCR technology, 20% were using the standard methodology, 14% were using some kind of convenient media or chromogenic type of media, 5% were using lateral flow immunoassay, and 6% were using other immunoassays. So this gives us type of 11% of the total market. The reason for it is that in the US, most large and medium kind of companies are sending their samples for pathogen to outside laboratories. And the outside laboratories have the required equipment to do the PCR technology. And that is what contributed to the high number of people using PCR technology, because the molecular methods require higher technical skill and better equipment. And in order to be cost effective, they need to use, you need to use many samples a day, and many laboratories have 1 or 2 or 5 samples a day, at which point PCR technology is not cost effective. However, a similar study done internationally found that only 20% of the people are using PCR type of technology. So my kind of final thoughts are that new methods are not being adopted as fast as developers like myself have envisioned. I thought that once a method gets AOC approval or ISO verification, that the method will immediately be adopted by the marketplace, but that's not the case. And a contributing factor is that new methods are being added very frequently to the marketplace. And it is both a blessing and a curse because of the variety of methods. It creates a confusion in the marketplace, and there are too many choices, making it very difficult, I think, for people to decide which method to use. And because of those new methods coming so frequently, many people are waiting for the best method to come, yet to come. And always there is a need for a faster, more accurate method, and more importantly, one that is like the multiple pathogens, that you can do one thing in all of them. So I think it would be very interesting to observe where the pathogen testing will bring us in the next decade. So thank you for your attention, and you can join us both on LinkedIn and in our website to get more information about food safety and food microbiology.
Thank you. Thank you very much, Dr. Eden. The presentation was very applicable and valuable for the colleagues in my office. I hope they are here, and we are waiting for any question.
I've got a question if you want. So the problem with these modern tests, I call them, is the number of false positives. You know, you can only go to look at the PCR method for non-0157 E. coli. So do you think the false positives are the real reason that people don't like the new technology? As you said, they like to have established.
I don't see really that the latest methods that are there have a super high level of false positive. Obviously, if you get a positive, you have to re-verify that you really have the organism present in the sample. But the fact that if you get in 8 hours answer, it allows you in the same day still to put the samples for further testing. And by the next day, you can have a final answer that is verified. So it is a huge advantage over the standard method.
And Dr. Eden, this method has FDA approved?
Oh, not FDA approved, because in the US, FDA does not approve food type of methodologies, but they all have AOC and ISO type of validation, I would call it, not approval because they don't approve, but they validate the method. So they went through international multi-laboratory type of collaborative studies that showed that they are effective in detecting the pathogens.
Thank you. Any question?
And Dr. Amini, you have microphone. If you have any question, you can ask.
What? I didn't hear the question.
Dr. Amini?
I hear you, but can you repeat the question, please?
No, one of our participants, Dr. Amini, has raised his hand to ask a question. I'm giving him a microphone, but please accept my request to activate your microphone.
Yes.
Yes, now we have your sound.
Yes. Thank you very much, ma'am, for a nice presentation. And one of your slides, you have told us that in a single test, we can detect multiple pathogens. How is it possible to differentiate them? Is it possible to say that, for example, this E. coli or Salmonella are specifically been isolated, or there's sometimes this other non-pathogen also might be in combination with those? How can we separate them and differentiate them on this basis? Thank you very much.
Yeah. The Cosmo ID is a company that have developed this technology and software that is used in order to separate. And I don't know exactly, because it's proprietary, how the software works, but basically it takes the fragments of DNA and it looks for certain sequences of DNA to be present in the sample in order to decide that a certain pathogen is there. The advantage is obviously that you can do multiple pathogens in the same sample, and they have demonstrated it, but it's not totally a done deal. That they have not done really validation of the method enough to say, yes, it works. It does show that many people are working, and that is what's coming in the future, more so than we have really today a method that works.
Yes, thank you very much, man. Thank you very much for your answer. Thank you.
And thank you. I have a question, Dr. Eden, about multi-DOF and Vitek. Could you please compare these two devices with your devices?
Which two devices?
Multi-DOF, an instrument for detection of pathogen bacteria in food.
I'm
You know, multi-DOF biotyper.
By whom?
Multi-DOF biotyper.
I'm not familiar with it, so.
Okay, thank you.
Yeah.
Thank you very much. And any question we have or not? If we have not, I would like to thank Dr. Eden. Thank you very much for your participation, for your valuable presentation. Thank you, Eden. Thank you.
I want to unshare my screen, right?
Yes, could you please stop the sharing?
Where is it?
Do not worry, I stopped your sharing.
Okay.
Thank you. So I would like to invite Professor Kate Warriner. Dr. Kate, are you here?
Yes, I am here. I'll just share my screen and see how it goes.
Okay.
And I'll just go into presentation mode.
It's okay.
I think it will be in a minute. Oh. All right. So can you see the full screen or the presenter mode?
Yes, it's okay.
Oh, so you're good. Are we good?
Yes, please.
I'll take that as a yes.
If we would like to introduce yourself first and then start your presentation.
Yeah,
Thank
sure.
You.
So my name's Keith Warren. I'm here in Canada. Nice picture of the Department of Food Science there in the corner, which is approaching fall. I think we're about a few weeks away from that. So I've been here since 2002. I came from the UK, where I did my undergraduate degree in food science at the University of Nottingham, then went to a PhD in the University of Aberystwyth in Wales, and then came over here in 2002. And our sort of research is focused on food safety, and more applied food safety. So we develop interventions, we look at pathogen plant interactions, human pathogen plant interactions, emerging areas, and a bit like the last talk, we actually do some diagnostics, which is interesting. But what I want to talk to you today about is something we worked on for about 15 years and got commercialized by Paul Moyer and Cleanworks about 5 years ago. And it's to do with a decontamination method, which we designed for fresh produce, but then went on to decontaminate lots of other things. So the method's based on what we call hydroxy radical in the vapor phase. And as the name suggests, it's literally a vapor of these hydroxy radicals that can decontaminate surfaces rapidly without causing changes. And what I'll do at the moment is not so much go into great detail, but just tell you how we got there and what the applications are and where we're going in the future. So the science behind it is very simple. It's been known, I would imagine, since 1860, when Fenton, he actually mixed hydrogen peroxide with a bit of ferric chloride, and he generated this strong oxidant. And these strong oxidants were actually hydroxy radicals. And hydroxy radicals are the most oxidative species known. So in nature, we see them in the atmosphere where they clean up pollutants. We see them in young stars, and they are literally very powerful, but very short-lived. So the half-life is less than a few milliseconds. But with those radicals, they're very potent in terms of antimicrobial and also degrading compounds. And the thing about them is that once they've reacted, they just convert back to oxygen and water. And if you get the conditions just about right, you can actually start making hydrogen peroxide. So it kind of sustains itself. But that's the basic science: UV, hydrogen peroxide, and we just mix some ozone into it as well. So this sort of reaction, as I said, isn't new. Fenton developed it as a chemical reagent to look at oxidation of carbohydrates and whatnot. And since the 1960s, it's actually been used in water treatment to actually degrade pollutants that wouldn't ordinarily be degraded by normal wastewater treatment. So organics, pesticides, dye, phenols, all these, and it's coming increasingly. And again, the principles are exactly the same. You have UV, you have ferric ions, which convert the hydrogen peroxide to these hydroxy radicals, which then oxidize the treatment. So the thing is that when we went into the vapor phase, we didn't really know what to call the process. So we did actually look at, well, advanced oxidation process, because then people could relate it to water treatment. But it was in the vapor phase, not in the aqueous phase. So we thought, well, vapor phase, advanced oxidative process, but that didn't seem good. So reactive trioxidane process. So trioxidane is this sort of free radical form that's very transient. And so RTP, or hydroxy radical plasma, because in a lot of ways, this process recreates gas plasma, but much cheaper, much simpler. Gas plasmas are all fancy with the glowing things, but the trouble is they're not very practical due to the need for high voltage and whatnot. So we haven't got a name for it yet, so we'll just go by those three names and we'll see what it is. So as I mentioned to you, we actually worked on this for 15 years, and we're looking at different produce. We just got hydrogen peroxide vapor, we got UV lamps, UVC lamps, and ozone generating lamps. And we started with apples because that's what Paul Moyer's business was, apple processing, or candy apples to be specific. We had this sort of basic unit, but it was very effective. So one of the big concerns here in North America, and I'm pretty sure everywhere else, is Listeria Monocytogenes. We get more recalls of Listeria than any other pathogen, but Salmonella is close behind it. So the thing was, using these treatments and the hydrogen peroxide concentration we vapored into the reaction chamber, basically that serves as a source of hydroxy radicals. And not unexpectedly, the more the concentrated hydrogen peroxide, the more radicals, the more log count reduction on the surface. But because the radicals are in the vapor phase, you could actually even go into the subsurface. So you actually got a decline in Listeria levels within the apple itself. So a very effective treatment in that respect. So one aspect is about Listeria pathogen reduction, but another big thing at the moment in North America is pesticides. So chlorpyrifos was just basically outlawed almost by the FDA in the US just last week. And there are issues, obviously, because they get into the environment, they cause issues. Consumers don't like them, and we've got a top 10 list of produce with pesticides on. But using that process I've just described, and we inoculate spike apples with chlorpyrifos and boscalade, which are the two common ones, you could get easy degradation due to the hydroxy radicals. So a very effective process in that way. So when we developed this in the lab, we were wondering, saying, well, I think the biggest people who will come to our door would be the leafy green people, because we've had so many outbreaks with lettuce and things like that. But oddly enough, it wasn't. It was the citrus lemon producers. So you don't associate lemons with many foodborne illness outbreaks. But the reality is that there was a recall to do with Listeria due to contaminated lemons. And this is the thing about North America, well, US, should I say, is that they've got this zero tolerance. So anything with Listeria on that's kind of ready to eat, like a lemon would be, is recalled. And there's been peaches and all this. Anyway, so the thing with lemons then is that if you overcook them, as in over-treat them, you get this sort of browning, this sort of sunstrike, and we've got other names. And what this browning is caused by is when the fruit is ripening and is facing the sun on the tree, it develops these polyphenols. And what the oxidative AOP process does, or the vapor phase process does, is bring out this, it like brings this sort of brown up. So you can't just apply the standard treatment, you have to kind of optimize it. And we optimize it using response surface methodology to look at the effect of UV, hydrogen peroxide, and ozone. And we came up with a magic formula. And based on that magic formula, CleanWorks, which I said was developed by or created by Paul Moyer, which was commercializing the technology, built this 16-foot line. And using the optimized characteristics, we could get lemons which were decontaminated without the browning, which was the sort of issue. So when we had this sort of large unit installed in California, we had to validate it. And as you've probably guessed, not many producers like bringing Listeria into a plant to validate the unit. So what we did here is actually we used a surrogate, and a good surrogate for this treatment is Lactobacillus fructivarans. So it's a very simple process, is that we get the lemons, we wash them, because usually you've got a bicarbonate coating on, which kind of interferes with the free radicals. We inoculate them, we dry the lemons, we put them in, then we recover, and then we count them. But using this method, what we demonstrated was that the process does cause about 4 log reduction of Listeria, which isn't bad considering washing doesn't do that much. And a unit like this, because the treatment is so rapid, can do 52,000 cases of lemons a day. And Fanny, who was part of us, is part of the Sunkist group. That's Paul Meyer, who was the CEO of CleanWorks. So very effective process, can be scaled up, and there's been obviously innovation since then. And one of the innovations is to do with frozen berries. So recently, in North America again, we've had this multiple recalls with frozen fruit and vegetables. And, people argue, saying, well, no one's going to eat frozen berries and frozen broccoli. But yeah, people actually do. And, freshly frozen berries. So you have to decontaminate them, because they actually trace some Listeria back to the freezer, because you think, well, if we decontaminate the berries before they go in the freezer, we should be good. But no, there's been multiple occasions where Listeria has been actually found in freezers, not growing, obviously, but still there. So it's a challenge, isn't it? Because you've got to keep the berries frozen, you can't change them. And CleanWorks came up with this unit here, which I've got a video of, and there's a golden rule, is never play videos in a presentation, because they always go wrong, but we're going to see if it does work. Oh, I think my sort of theory that you should never play videos in a presentation has just come true. But anyway, so what I can tell you is that, oh, hang on, something's gone wrong. See, that's what you should never do. I'll try once more. I don't think it's going to work, but oh, it's all broken down now. So I'm just going to go out of the slideshow and go back in if that's all right, because I'm seeing two things at once. Okay, so we're not going to get there, are we? So with a frozen berry line, as I mentioned, it's exactly the same process that I've just showed you for the lemons, but we have what we call an intermister. And this intermister kind of mists the hydrogen peroxide rather than a vapor. And so by doing so, you can keep the berries frozen, but still support a sort of 2 to 3 log reduction of Listeria. Oh, here we go. So never play videos, there's a lesson. So we've tried it on lots of different produce type, avocado, apples, cantaloupes, and these are only a selection, leafy greens. And this is a standard treatment, but if you want to improve it, you can always optimize it. And you optimize it by the hydrogen peroxide concentration, ozone concentration, even the orientation of the produce. So a very effective method with no negative effects. More recent work, and this is never going to work, is our microgreens. And again, this was a video of a microgreen decontamination whilst it's still on the tray. This is pre-harvest. Oh, you never know, it might work. Oh, we could be lucky here. So yeah, there we go. So what we have is UV, the hydrogen peroxide vapor, ozone being introduced. And with microgreens, you can't really wash them post-harvest because they spoil so quickly. So you would treat them pre-harvest and then harvest some after that. And you get about 3 log reduction. So as you can see, a very rapid process, relatively simple, much simpler than gas plasma and effective. So that's one thing, but because the treatment has low water, you can do low moisture food. So over the past 5 years, we've had big issues with low moisture food, especially with salmonella. And we've done trials with dates, cherries, blueberries, pistachio, salted pistachio. And it is, again, you have to optimize the treatment, but because these are water sensitive, much simpler than irradiation, much more cleaner than using chlorine dioxide or other fumigating agents, and fairly effective. And in addition to that, we've done work currently on shelled eggs. And what's interesting about shelled eggs is that there have been issues with salmonella before. And what they use presently in the industry are washes. So they'll wash them in chlorine, and they've got the temperature differential. Very laborious process, very damaging process, because in North America, this trend of eggshells getting thinner, you know, for some odd reason, so you get a lot of wastage. But they're absolutely ideal for the gas phase hydroxy unit because we can decontaminate shelled eggs with only 1% hydrogen peroxide with a 10-second treatment. So you can imagine how many eggs you can do. And it's again just getting the right hydrogen peroxide ozone concentration, not so much, but UV intensity right. So it's a very effective treatment. That I was quite surprised at eggs, given that they're very porous, where the pathogens can reside within the pores. In addition to that, we've got another process based on not hydrogen peroxide hydroxy radicals, but chlorine radicals. And the reason why we have to use chlorine radicals on protein like chicken wings and fish is because of the high catalase content of the meat. So if you put hydrogen peroxide down, it fizzes and bubbles, looks impressive, but it doesn't do anything any good. So we use the chloride radical, which is generated in a similar manner, you know, by UV and ozone. And you get around about 1 to 1.7 log reduction of Listeria, you know, over 90%. And this is critical because, as I mentioned to you, in North America, we're getting really tight on pathogen carriage, even on raw poultry, which you say, well, yeah, chicken has salmonella, but you know, this is an issue with salmon, which is obviously can be eaten raw, if you like that sort of thing. Again, you've got to be able to have a process. And correspondingly, they've been using hydrogen peroxide, peroxyacetic acid, which both are very detrimental to the product because it causes shrinkage. So another effective treatment. But during the pandemic, I know we're not allowed to say anything to pandemic, we actually pivoted the process from decontaminating fresh produce to decontaminating personal protective equipment. And we're going to try another video. So in this video, we were looking at N95 masks, so which are basically a very short supply back then. And so we used a similar treatment, and we also developed a chemical indicator, which was based on conducting polymer. So because what you have to do is verify that the thing's sterilized, but if you don't, people say, well, I'm not going to wear that. And you'll notice maybe if eagle-eyed, it changes color from colorless to black, and that tells you it's properly sterilized. And we've done face shields and goggles, and it's applied in healthcare facilities for this reason, just to extend the use of these masks. And unlike what they used to use, the sort of older methods, which are methods of using, they have to be sent to a central hub, treated with hydrogen peroxide vapor, and it could be 10 days before you get your mask back. And even then, you don't know whose mask it is. But with this treatment, you literally can decontaminate your mask whilst you're washing your hands. So very good in that way. So what an unusual, not unusual, but a sort of interesting observation. We developed this process for inactivating pathogens on foods and on food contact surfaces. But we started to realize that we're actually extending the shelf life of produce as well. So lemons, for example, you can stop them spoiling quick, but the biggest impact was on strawberries. So we all know we buy strawberries, we take them home, and a few days later they go moldy with botrytis. But what we found is that if you treat them with the AOP process, or hydroxy radical process, you get up to 15 days extra shelf life. And this goes beyond just microbial reduction. This actually alters the physiology of a plant. It occurs with peaches. One of the biggest impacts I saw with avocado. So this was done down in California again, the avocado capital of the world, I suppose. And we treated them with the clean flow system, and with controls which weren't treated, only about 3 fruit actually made it to the end of a 10-day shelf life. But with the clean flow treatment, totally good. And again, beyond the radical just inactivation microbes, it's altering the physiology of the berry. Now, another example was golden berries, which go under different names, cherry, I think tree cherries or something like that. But the thing with these golden berries is they spoil really quickly. So you have to treat them, and you can't wash them because it destroys them. But we did a clean flow treatment, and this was after 30 days. As you can see, the controls are all molded up. The clean flow treated are looking good still. Not sure how they taste mine. Yeah, I never tasted them even on the controls. But anyway, so in conclusion then, obviously we've developed this process, which is, when you think about it, relatively simple. You're just using vapor, UV and ozone, and it can decontaminate a broad range of microbes from viruses through to endospores. No changes to surface, is very rapid. You're reducing the water usage. You're extending shelf life. It can be continuous or batch, small or large, as you've seen. So it's a very versatile piece of equipment. And it really has had an impact on the industry, and there's been multiple awards. Paul got an award as Innovator of the Year. Madia, who's my postdoc, who actually did a lot of work, and as you say, she's your fellow countryman woman. She won award for the commercialization. We won award at the IFAP for Food Safety Innovation of the Year, etc. So it is getting sort of foothold in the industry. Obviously, industry, a bit like the previous speaker talking about biosensors, are very reluctant to adopt new technology. But, you know, as forces, regulations, and proven efficacy of a treatment comes up, we're definitely finding more application for sure. So thank for that, and I'll stop sharing, and I'll try and answer any questions anybody might have.
Thank you, Dr. Professor. Kate, now we are waiting for any question. No question?
Well, Bahram's got his hand up again, so if you want, yeah.
Dr. Amini,
Yes.
you have your voice, please.
Yeah. Sir, thank you very much for your nice presentation. I had a question regarding these chemical substances which we are using as a disinfectant on the food products. Do we have any residue of these chemicals on the substances? And if it is so, won't they be unsafe for human health and for the environment also? Thank you very much.
Oh, no, thanks for that question. And you're exactly right. You know, if you think about chlorine, the chlorine disinfection byproducts that are produced, like chloroamines and things like that, if someone said we're going to use chlorine as a sanitizer today, they would ban it because of that. Now, with our system, you don't get any residues because it's so rapid. And there have been questions, especially in the gas plasma world, about the radicals that are formed. Are you producing something that's actually more toxic than the original product? And with gas plasma, because the treatments are so long, they do generate these sort of these products that people say, well, I'm not sure if that's actually safer than the actual original pesticide. But in our treatment, we've had different studies. One with apples, with the mask treatment and other things like that. There's been no residues detected. And we've even looked at the sort of residual washings, if you want to call that, coming out of a machine, the waste solution. And again, there's nothing there. So it's one of those, and you know, these sort of treatments are fairly effective, but you are right. I think one of the other hurdles you come across is people say, well, trioxidane, that sounds a bit dangerous, but trioxidane comes and goes. But certainly, yeah, compared to ours, no radicals, but definitely other sort of treatments, even chlorine dioxide, there's been a lot of issues with toxic byproducts.
Yeah, thank you very much, sir, for your kind attention. Thank you.
Thanks for the question. Good.
Thank you.
Okay, thank you, Professor. And now we have a short break. And after that, I invite Dr. Safayan for her presentation. Thank you very much. Thank you. Thank you very much. Dr. Safayan, could you please start your presentation? Thank you very much. You are on mute. Please unmute yourself. Thank you.
It's okay.
Yes.
My name is Sheila Safayan, and Associated Professor Islamic Azad University. My speech about detection methods for foodborne pathogen viruses. Good evening, ladies and gentlemen. Hello, everybody. It's my honor to be here. First of all, I would like to thank Professor Feyzabadi, Professor Rahimi Fard, and Dr. Abdullahi for warm supporting in order to hold this conference. I would like to speak about detection methods of food-borne pathogen virus.
Sorry for the interruption. Sorry. Could you make it full screen? In full screen mode.
Yes.
No, it is
Okay.
still in the PowerPoint. It is not in full screen. At the top menu, beside animations, you can, yes, now it is in full screen. Thank you. Please continue. We can see your slide in full screen mode. Thank you.
Introduction. Numerous viruses can be found in human intestinal tract. Food and waterborne virus that can be divided into 3 disease categories. virus can cause gastroenteritis, such as adenovirus, astrovirus, rotavirus.
Sorry, we lost Dr. Safayan.
Dr. Safayan?
Dr. Safayan, if you can hear us, I send you request to start your video and your microphone.
You sent for me. It is, this is Shima Safayan.
Shima. Shima
Yeah.
Safayan. No, Sheila. Dr. Sheila Safayan.
Yeah.
Where is Dr. Sheila Safayan?
Okay, I guess she got disconnected.
Please wait. Yes, we are waiting for her. No problem.
Oh.
Has a problem for connecting us, so I would like to start my presentation till Dr. Safayan would be ready for the next after me. Okay, I'm sure.
Dr. Rahimi Fard, we can see your slide, but it is in PowerPoint. Please make it in full screen mode.
Is okay or not?
Please make it in full screen mode. On the up, beside animation, you can see a slideshow menu. Yes, beside that. Yes, at the top. Beside home, insert, design, transition, animation, and a slideshow.
Yes,
yes.
From
From
the beginning,
the beginning, at the left side. Yes.
Thank you very much. Okay. Hello, everybody. Again, it's my honor to be here. With my colleagues, I would like to speak about the nutritional support in COVID-19 disease. As I mentioned in the last panel, excuse me, I want to. How can we close this? Excuse me. Okay. As I mentioned in the last panel, nowadays close physical communication in worldwide population is unavoidable, like the virtual online internet communication. We shall actually consider a union country and believe in its existence instead of separate national countries. I call this huge country Earth. Due to severe contagious of COVID-19 and above mentioned fact, mostly all people catch this type of flu by this agent, but based on the case conditions, the disease manifests in different signs and symptoms case by case. Therapy and rehabilitate and regain optimal health status for survived COVID-19 patients in COVID-19 epidemic that broke out in Chinese Wuhan in late 2019 proved the important role of the nutrition and the immediate implementation of caloric and protein balanced nutrition care in viral diseases. The Chinese have published their recommendations, including principles for diagnosis of nutritional status along with the optimal method for nutritional supply, including guidelines when to introduce education approach, oral nutritional supplement, tube feeding, and parenteral nutrition. They also calculated energy demand and gave their opinion on proper monitoring and supplementation of immune nutrients, fluids, excuse me. Wait a minute, excuse me. Fluids and macronutrients intake. Nutritional treatment of patients consists of intake of liquids, macro and micro adequate the immune system and nutrients to enhance, fulfill vital requirements. Nutritional approach should be an important section of therapy in patients with COVID-19. Immune system function plays an important role in the prevention of respiratory disease, including coronary heart disease. Compromised patients with diseases such as diabetes and lung diseases, heart disease, and malnutrition are more affected with coronavirus. Lack of food intake and deficiency of vitamins such as vitamins C, A, and D, and compromised immune system increase the risk of disease. Daily consumption of vitamin C sources such as vegetables and salads, fruits containing vitamin C such as orange, tangerines, lemons, kiwi, and vegetables containing vitamin C such as cabbage, cauliflower, turnips, green peppers, and bell peppers, which they are good food sources of vitamin C, is recommended. For example, daily consumption of 1 orange or 2 tangerines provides the daily requirements of vitamin C. Carrots, squash, and dark green vegetables such as spinach, beet leaves, and dark lettuce leaves are also good sources of vitamin A. In general, in order to prevent disease and strengthen the immune system, daily consumption of 3 units of vegetable except starch vegetable and at least 2 units of fruits is recommended. Children under 5 years of age, pregnant mothers, the elderly, and patients taking corticosteroids are following precautions to prevent it. Use vegetable or salads with lemon juice or fresh orange juice every day. Use carrots and squash in daily meals. Consumption of raw onions with food is recommended due to having vitamin C. Avoid eating sausage and other fast foods and fatty and heavy foods. Using protein sources in your daily diet, such as legumes or eggs, is important. Lack of micronutrients such as iron and zinc weakens the immune system. Use the food sources of these 2 micronutrients, such as legumes, and as a suitable substitute for meat, milk, and dairy products, green leafy vegetable, and more nuts. Avoid eating foods that are not well cooked, such as eggs, kebabs that are not well cooked. Avoid eating food and liquids in places that are not safe. If you have symptoms of a cold, use watery foods such as soups and stews with fresh lemon juice and warm liquids. Use wheat germ, mung bean, and clover in your daily diet. Consumption of foods containing a variety of antioxidants, such as dark chocolate, spice such as turmeric, is recommended. Remember that balance and variety in your daily diet and ensuring that you get enough micronutrients and protein from your daily diet play an important role in preventing diseases such as coronavirus. In addition, having enough sleep, taking probiotic products or supplements is recommended. And finally, avoid stress and physical and mental pressures. Okay, thank you. I think Dr. Safayan is ready for your presentation. I invite Dr. Safayan. Are you ready?
Yes, I'm ready.
Okay.
Sorry for interrupting. Again, hello. I will start from beginning. My title is the Detection Method of Food-Borne Pathogen Virus. Introduction. Numerous viruses can be found in the human intestinal tract, food and waterborne viruses that can be divided into 3 disease categories. Number 1, virus caused strains such as adenovirus, rotavirus, and calciviruses. Number 2, fecal orally transmitted such as hepatitis A.
Sorry, it seems that we lost Dr. Safayan again.
Dr. Safayan, I think your internet has interruption. I apologize to everyone here. I think she couldn't continue to her presentation, but no worry, we can share her slides with all of you next, and we will tell you how you can use all of the slides of all of the speakers. If there is any question, please type in the chat. So Dr. Ruth Eden is here, Professor Kate is here, and we can, and also Dr. Safayan. So we can be together for, I think, for 10 minutes later. Is there any question or any suggestion? Thank you very much. I think there's no question.
Dr. Rahimi Fard, we have one question in the chat box.
Do you have any supplements for lactose intolerance patients in Iran? If yes, please state the name. Yes, we have, but I think one of the Iranian company, I think one of the Iranian company, I search and Bita Rahmani. Yes, I will tell you. We have lacto. I don't know the exact name of this company, but we have peel for the patients that has lactose intolerance. We have this supplement, yes. And one of the Iranian company produce it. I tell you next. And another question? Professor Kate has written that vitamin D has been shown to help COVID symptoms, although evidence is not definitive. Yes, exactly. The vitamin D is one of the best supplements for prevention and also better to remove the symptoms of COVID-19. It's proved. That's lovely. If you can share with me, thank you. Yes, absolutely. I will tell you this supplement. Please tell me your phone number here. I will be touched with you. Thank you. And do you have any suggestion or any question? You can thank you. You can use the slides and the presentation in the site of this Congress. Dr. Abdullahi and their colleagues will put them there. Now, if we have any question, I thank the dear speakers that are here yet, that are here, and I thanks for all of your audience, and hope you safe and health. And I hope see you in the next panel in the 13th of September. The panel, the second panel of food microbiology in this Congress. Thank you very much, and bye-bye. Bye-bye.
Thank you, Doctor. Dr. Abdullahi, please share your clip.
Puratab Pharmaceutical Group, with more than 2 decades of activity, is one of the leading holdings in the field of health in Iran. This company, as the first and largest importer of pharmaceutical supplement company, has been able to play a significant role in maintaining and promoting public health. In this regard, probiotic supplements with advanced formulation and under the license of Swiss Phytopharm have been included in the product portfolio of this group. The most important features of probiotic of this company are: the highest quality of our products has always been a priority for the Probiotics group. Besides, we are committed to continual improvement. In this way, introduced several kinds of probiotic products into the market with outstanding properties such as specific strains for different target groups, lyophilized strains which are deposited in international culture collection, alive in sufficient and high numbered products during shelf life, fortification with probiotics, vitamins, essential elements, products information, Emulact, Lactine Fent, Kiribium plus D3, Kiribium plus Inulin, OptiKits, Bioget, IB-Syn, Megabiome, Pylori Tect, Lactoget, Genelect, Probiotics from Vivatune brand. Be balanced.
Thank you very much. Dr. Safoyan, unfortunately, we can't have you and your presentation, but thank you for joining us again. Thank you for share your slides. Thank you very much.
Thank you.
Thank you. Bye-bye.
Yeah.
Goodbye, everyone.
Goodbye.
Bye-bye.